Um, all right. Hello, everyone. Welcome to another session of our evolution seminar series. Today, we're going to take a dive into the world of phylogenetic networks as we're joined by Professor Claudia Solis-Lemus from UW-Madison's Department of Plant Pathology and the Wisconsin Institutes for Discovery. So please join me in giving a warm welcome to Professor Solis-Lemus.
Okay, can people hear me well? Yeah, okay. Yeah, thank you. So I want to start just by saying that I have a lingering cough, that I'm not contagious. I had a cold like 2 weeks ago. I've been tested. I'm fine, but the cough refuses to leave me. So if I just cough every once in a while, I just know that you are safe, but also, I'm just like, can't stop it. I guess cough is part of my life now. So anyway, yes. So I wanted to give like an overview. It's been a while since I've given a seminar here, both in evolution and then at the university. So I wanted to give more like an overview of different topics that we're going like investigated in my lab. Perhaps not getting super deep into any of them, but we can dive deeper once there's time for the discussion or the Q&A if there's interest. And my lab has three main themes. There's phylogenomics, which is the study of evolution, machine learning, and then microbiome. And I will focus more on phylogenomics today, but I will briefly talk about machine learning as well, and very little microbiome by the end. Okay, so like I said, in phylogenetics, one of the main goals is we want to reconstruct the tree that represents the evolutionary history of some group that we're studying. So for example, here we know that the tree has internal nodes that represent speciation events. So we know that this node represents a common ancestor for these two fish, and then so on and so forth until we reach the root that is the common ancestor of the whole group. When we have a different type of biological process called hybridization, for example, this is the case where we not only have speciation events where different lineages arise from ancestors, but we now have the case where these separate lineages now hybridize and create a new species. And that's the case, for example, of this hybrid speciation. We represent that mathematically with a network where we now have another type of node here in green that we call it a hybrid node. So this node, it represents some form of hybridization event in which the genetic material that is beneath this, well, I mean, this is horizontal, but that is descendant to this node has two different origins. So one of the origins is coming from this green arrow, and the other origin is coming from this other path. We use the term hybridization, but we don't mean just hybridization or hybrid speciation. This type of process could actually be any form of gene flow. So it could be horizontal gene transfer, migration, recombination, reassortment. There are many ways. There are many biological processes that have a signal that is not well represented by a tree. All of those are represented the same way with a network. This is to say that when we're using a network methods to get a network, we do not know the biological process just by inferring this network, because this network, the exact same network, could be originated from different types of biological processes. We need other information to know if this pattern that we see is coming from hybridization or introgression or horizontal gene transfer. We need to have other information that the method cannot give us. The method will give us, there is signal here of something that's not really tree-like, but what is the explanation for this event? Well, the method doesn't know. And the bulk of what I do is develop methods that will infer these phylogenetic networks from sequence data. So we have whole genomes or multiple loci. We estimate gene trees for each of these locus, and then from the collection of gene trees, we estimate a phylogenetic network. When I joined the field of phylogenetic networks, there was a state-of-the-art software called PhyloNet that would reconstruct phylogenetic networks from samples of gene trees. And this software was very accurate, but it was not scalable at all. For example, if you had anything bigger than 10 species, you could not run, or 10 taxa, you could not run PhyloNet. I mean, you could try, but it will take forever to finish. So this is a simulation study where we have a set of just 10 taxa, 2 hybridization events. And then this is the line on top is the running time of PhyloNet. So it just really more than exponentially growing with the number of genes. So this was not a scalable process. So when I started working on this with Cecile and Nea as part of my PhD, we wanted to create a tool that was scalable. We wanted to be able to build networks that are much, much larger. And we created this method called SNAC, that stands for Species Networks Applying Quartets. This method, as its name suggests, is a quartet-based inference. So we are dividing and conquering rather than evaluating the likelihood of a big network that has 10 taxa. We split up the likelihood in sets of 4. So if we have a gigantic network that has 50 taxa, for example, evaluating the likelihood of this big network is really, really computationally expensive. So what we're doing is we are extracting sets of 4 and evaluating likelihood only on the subset of 4. And then we repeat this process for every subset of 4. It might sound that we're doing more work. Instead of doing just one big computation, we're doing n choose 4 small computations. But it turns out that just even by doing a lot more computations, the fact that they're much smaller, the method is much more scalable than the original likelihood approach. So we implemented this in a Julia package that's called Phylon Networks. I don't know if people are familiar with Julia language. It is similar to R or Python, but it is much faster and more efficient, in my opinion. Actually, that's it in general. It's true, 100% true. And it's also very easy to code. It's a high-level language, so you do not need to be a computer scientist to do efficient code in Julia. Anyway, we implemented this package, and it was much more scalable than PhyloNet. So here we are showing that the running times of PhyloNet and SNAC on 4 cases, the largest was still very small, just 15 taxa, 3 hybridization events. We couldn't run PhyloNet on this setting at all, and we could run SNAC on all of these settings. In fact, SNAC, we're able to run even networks that have like 50, 60 taxa, which still doesn't sound as much if you think of like the large scale of tree of life, but it is massive number when you're thinking of phylogenetic networks, just comparing to what we can do before that was at most 10 taxa. So one of the advantages of SNAC compared to previous network methods was scalability. We were able to reconstruct larger networks that we couldn't do before. And the other thing, the other advancement with SNAC was identifiability. And to tell you about identifiability, I like to use this cartoon where all of these processes basically are trying to find a network or a tree that maximizes a function. And I always describe this process the same way. It's like we are trying to find the tallest mountain in a landscape with our eyes closed. So that is what the software is doing. So it's searching the space by taking steps until it reaches a top or what it thinks is the tallest mountain. And that is where the maximum likelihood or the maximum pseudo likelihood network will be. So this is a case where our function is well defined. There is a clear maximum, right? This is a case of a model that is identifiable. But imagine the case where all the trees or all the networks have the exact same value for our function that we're optimizing. Then we don't have any information from the data, from the model to identify which is the best network or which is the best tree. This is a case of lack of identifiability. So we really want to make sure that the methods that we use are identifiable, or in other words, allow us to identify which is the best tree, which is the best network for our model and our data. So we really wanted to investigate whether we could identify the best network with the data that we had. And we show that whether a hybridization event was identifiable dependent on the number of nodes that were in the hybridization cycle. So I'm drawing the networks a little strangely here as unrooted. And the hybridization event creates a cycle in the tree. And this cycle has certain number of nodes. So for example, this cycle has just 2 nodes. This cycle has 3 nodes. This has 4 nodes, 5 or more. So what we found was that if the hybridization event creates a cycle that has 5 or more nodes, then it is always identifiable. We can always detect a cycle that has 5 or more nodes, which mathematically is okay, but biologically does not make a lot of sense. Because if the cycle is really big, that means that the donor and the recipient of gene flow are really far apart in the tree for them to create this really big cycle. Donor and recipient hybridization events that are closely together will create smaller cycles, hybridization cycles. It turns out that these cycles, 2 or 3, are the hardest to be detected with the models that we have and the data that we have. In fact, gene flow between sister taxa is almost impossible to detect from what we have. So we can always try to detect cycles that have at least 4, 5, or more, which are not as common as we would expect them to be. But anyway, I think that it's still important to do this study of identifiability to know what are the limitations of what we can detect or not from the data that we have. So the direction that my life has taken since I started doing phylogenetic networks has been, we're trying to push the limits of what is it that we can infer. I told you with SNAC, we can estimate networks that have like 50 taxa, still not enough. No one is super happy. People want to build networks with 100 or thousands of taxa. We're at 50, so we're still trying to push that limit. We also want to be able to estimate more complex and complex network. I will tell you in a second why. And then test the limit of what we can detect. So I will briefly highlight some of these themes. So I told you that SNAC is faster, but it's not really fast. So if you want to estimate a phylogenetic network, you need to be patient. So for example, one case where that we had like 25 taxa could take like 3 days to run. So it's not really fast. You know, it's faster than PhyloNet. Actually, you couldn't run PhyloNet in that example, but it's not really fast. So one of the things that we're trying to do with smarter ways to parallelize computation of likelihood, to decide better ways to move through the space of networks when we're optimizing, we're able to get even 6 times faster running time just by smart computation algorithm choices. So this is something that we're putting together into a new version of SNAC that will be 6 times faster than current version of SNAC if you have used it. So this is very exciting. But I think that there is also a bottleneck on how big of a network we can estimate in one go. I told you that the inference procedure is we're trying to find the tallest mountain in this landscape. And the landscape, as you have more taxa in your data, this really is a massive, gigantic space that we cannot traverse with any computational procedure. No matter how efficient it is, you cannot traverse it all. So there's going to be a bottleneck of how big of a network we can estimate, even with a fast algorithm like SNAC. So the next approach that we're investigating, and this is work by Nathan and Kevin in my lab, is let's try to break up the taxa into subsets, infer network in each of the subset, and then merge them together into a larger network. So this is the algorithm for what we're calling infinitely large phylogenetic networks. I'm not very creative with names. This is all Nathan that came up with the name. So we have this algorithm where we have some data, we're breaking up into some subsets. For each of the subsets, we build a phylogenetic network with SNAC or with any tool, and then we are putting them all together into this large network. And Nathan has been able to build accurate networks up to 1,000 taxa with this approach. So we think that it has actually very good promise. There is a limitation, and it cannot really infer very ancestral gene flow events. It can only infer more recent ones, but we are building up tools to try to infer those more ancient hybridization events. And another approach that we're taking to try to get scalable inference of phylogenetic networks is let's try to bypass the traversal of network space altogether. And Bella is a student that she graduated, and she came up with a tool that is it's an ultra-fast learning method to infer phylogenetic networks using algebraic invariants. And I'm sorry, I'm going to have to pause to explain a little bit of what algebraic invariants are. So for the case of 4 taxa, A, B, C, D, there are only 3 possible unrooted quartets that I can draw. So I can either put A, B on one side and C, D on the other, or A, C, and B, D, or A, D, and B, C. So we are all agreement that there are only 3 ways I can draw an unrooted tree with 4 taxa. These are all the 3 possible trees that exist with 4 taxa. These are the only ones. Okay. Each of them, I'm not going to get into details, but each of them have a probability of appearing in my data when the true species tree is the one that I highlight here. So if the true species tree has A and B as sister taxa, then I will see this quartet that also has A, B as sister taxa with this probability, 1 minus 2/3 e to the minus t, where t is the ancestral, the length of the ancestral branch that leads to AB. So intuitively, if this branch is really long, then any lineage that I get from A, or any individual that I get from A and B will have a lot of time to coalesce in this ancestral population. So it is very likely that I observe this split. So if t goes to infinity, this quantity, the e to the minus t goes to 0. So this probability goes to 1. Most of the time, I will be observing a gene tree that matches the species tree. Yeah? But on the contrary, if this branch is too small, if this ancestral branch that leads to AB is very, very short, then A and B, they don't have time to sort before reaching the C population. And there will be a lot of discordance. So I will very likely, with same probability, 1/3, 1/3, 1/3, I will be observing the 3 quartets in my data, in my gene trees. So again, you have to trust me. I'm not getting into the details of what these probabilities are coming from, just saying that from the multi-species coalescent model, if the true species tree has A and B as sister taxa, I have some probabilities of observing different gene trees in my data. And if I give some number to these 3 probabilities, I'm going to call the first one A1, A2, A3. Because they're probabilities, they need to sum up to 1. So A1 plus A2 plus A3 needs to be equal to 1. They're probabilities. We know that they need to sum up to 1. And these 2 quantities are identical. So I should also satisfy that A2 needs to be equal to A3. These 2 equations, as simple as they look right now, these are algebraic invariants. So algebraic invariance, as a summary, is just equations that need to be satisfied for the model to be consistent, to be really a true probability model. So if they don't sum up to 1, they're not probabilities. If these 2 are not equal, they're not really coming from the model that I want. So algebraic invariants are simply equations that the model needs to satisfy for the probability for the numbers to be coming from the consistent probability model. So these are what called algebraic invariants. Okay, so what Bella did is she defined the set of all possible probability algebraic invariants for a specific placement of the hybridization cycle. So if you have your tree and you say, okay, I'm going to place the hybridization cycle in some part of the tree, that defines a set of algebraic invariants. If this is the true placement of the hybridization cycle, these invariants need to be true. If I move to a different place, there's a different set of invariants that need to be true. So the placement of invariants, it defines a set of, the placement of the hybridization cycle defines a set of algebraic invariants that if they're true, then we know where the cycle needs to be in the tree. So instead of searching the space to find the tree that maximizes or the network that maximizes the likelihood, we just need to evaluate invariants. Are the invariants true? Then that's the correct placement of the hybridization cycle. So we just need to iterate over sets of invariants. Some invariants look really ugly. So I was showing you some of the simple and pretty ones, A1 plus A2 plus A3 minus 1 equals 0. But these are all some other invariants that we run into. We're not trying to understand what they mean. Maybe they mean something, maybe they don't. At this point, we're just saying, okay, if these invariants are true, they are pointing at where the hybridization cycle should be. So Bella's work was actually very innovative. It was the first time that algebraic invariants were used to place hybridization cycles in trees. And we dare to call this ultra-fast learning because we used Bella's tool, actually implemented in a Julia package as well that anybody can use called Phylodiamond, to build this phylogenetic network for canines. So, and actually this is a photo of my dog. And her DNA was not used to build this tree because otherwise, I mean, she's pure jackal. I think she would confuse the analysis completely. Anyway, so we used our method to build this phylogenetic network, and the phylogenetic invariance method took less than 7 seconds to build this network, whereas the second fastest, which was SNAC, took 20 times as long. So and again, this is just an artifact of, we're not searching the space of networks anymore. That takes a lot of time. We're just evaluating polynomials. That is a very fast computational procedure. So we implemented this in Phylodiamond. And give me 1 second. Sorry about that. Okay, so we are pushing the limits of what we can estimate, bigger trees and with different techniques. And then we also want to expand to more complex types of phylogenetic networks. So the original version of SNAC only allowed what's called level 1 networks. And level 1 networks are networks in which the hybridization cycles, they do not intersect. So this is a case of a level 1 network. So you have different, you can have multiple hybridization events in the same topology, but they cannot intersect. So they cannot share edges. If you see, they are pretty separate in the tree. This is a case of a network that is not level 1, because if you see these two cycles, they share this edge. Yeah. And actually, maybe I should be using the pointer for those that are in Zoom rather than, yeah, this edge. I just forgot about that. So right now, the current version of SNAC can only estimate the level 1 networks, but we want to extend that to broader classes of networks because there's no reason why evolution should follow a level 1 network approach. So in fact, one of the original papers that we use this data set for fish, this is one of the networks that they estimated. This is not a level 1 network. It has a bunch of hybridizations. They are all on top of each other. We cannot estimate this type of network right now with SNAC. So this is a schematic of network space. Here, each of the nodes represents a type of network, starting with trees. A tree is a type of network that has no hybridization events. There's level 1, the one that has been mainly studied by us. And then here is level K. We're extending to level 2 networks, just one tiny step forward. But there's a lot of different types of networks out there. So Kevin is a postdoc working with me, trying to extend SNAC into now level 2 networks, and it's really complicated. And I get really excited about network space. And in fact, I sometimes describe myself as a biologist of networks. So this is my organism of study. It's a network. I even came up with a scientific name, Grapho Networky. And this is a picture that I took of a network in the wild. You can imagine that there exist networks in this mathematical space that has all these different classes. So there are all these different species of networks, and some networks represent some types of biological process. Another networks might not represent anything, or they're just be mathematical objects that are not really connected to anything in biology. So one thing that really fascinates me is what of these networks, how are they connected to the networks that we want to get from biological data? How do we better interpret them, and how do we estimate them in more scalable and accurate ways? So this is the space of networks. All that we know now is just level 1 networks. This is all that we know. We really want to extend that to bigger classes of networks. So that is the other direction that I'm taking, just starting with level 2, but there's like a huge space of networks that we still want to explore. And I don't have time to cover, but we also did a extensive simulation study comparing the accuracy of hybrid detection methods like ABABABA test and HIDE. We found that some of these methods have a very high false positive rate, in the sense that they will find gene flow events where they are not. And this was the case more often for ancestral, like really ancient gene flow events, and also events, gene flow events that involve ghost lineages. So whenever you don't have one of the lineages sampled from your data, these methods will have a higher rate of false detection. And just to conclude this part of phylogenetics, I want to show, like, I'm a statistician by training. And one of the things that we'll find here from this talk that fascinates me is how do we represent things in a mathematical way? Like, we want to know, how do we represent something as numbers? Because the computer and statistics deals with numbers. So we want to know, how do we make that connection? And I'm collaborating with Julia Palacios from Stanford to represent networks into matrices. This is not the adjacency matrix, by the way, if anyone's familiar with adjacency matrices. This is what's called an F matrix that represents the ranking order of lineages. And by representing networks as matrices, then we can do any form of statistics that we want. We can calculate means, we can calculate variances, we can cluster networks. And Julia has done already all this work for the case of trees, and we are extending it for the case of networks. Similarly, Ivo is working on another way to embed trees into numbers, which what we call split weight embedding. So we're representing the tree, each tree as a vector of branch lengths, depending on the splits. And then we're able to cluster gene trees based on this embedding. We also developed a Julia package. We don't have a logo, but Ivo wrote it in the snow. This is how old it was. So last winter, he wrote it in the snow, follow clustering Julia package. The whole point of this, I don't know if I included a slide. No, I did not. Is if you have a sample of gene trees and you want to cluster them by the topologies, we can do that with this embedding. And this is getting a little bit now into machine learning. That is the next thing that I wanna talk about. But anyway, so some of the themes for my lab in phylogenetics is we want to keep building larger networks, more complex networks, and then trying to find that connections between the mathematical world of networks and the biological world of what is it that we want these networks to represent? What are the reticulate evolutions that we want to represent? And then some of the things that I'm getting into for the future is polyploidy, that I have been pretending up until now that is not a thing, you know? Like, oh, I didn't say that, but all the methods that networks, they assume you have single copies, you know? And if you're not in that case, you need to either throw out, you know, your data or find some other ways to get into the single copy world. I'm lucky that I have collaborators like Josh Tiley is a botanist that is trying to push me into biological realism, you know, despite my resistance. So one of the things that we want to explore is how to incorporate polyploidy as a process that to infer networks that account for incomplete linear sorting, hybridization, and polyploidy same time. And the other thing is pangenome. So Emil Gluck-Thaler, he's another collaborator, he's a mycologist. Like the whole notion of, I'm not an expert, but the question is, can we build better trees when we have pangenomes as opposed to having just alignments? That's one of the things that we want to explore. And the third thing for the future, and I put this emoji, machine learning, like I said, I'm a statistician by training. And I really never wanted to get into machine learning, if I'm completely honest with all of you. I never wanted to. I was like, you know, that is not for me. Some of the computer scientists should deal with that. You know, I'm gonna keep doing my likelihood optimization, you know, maximum likelihood, because we have theoretical guarantees, and I can go on a tangent about many things that I don't like about machine learning, but it's hard to, you know, to be the only one that is not doing machine learning. So there's all this pressure, you know, for students want to do machine learning, collaborators want to do machine learning, everybody wants to do machine learning. So say, okay, let's do some machine learning. And some of the first projects that we did, they were very traditional machine learning. And by that, I mean they were classification. Machine learning is very good at classifying things. We have all seen this example of classifying dogs or cats, right? Like classifying images of different cars. It's very good at classifying. So the very first projects that I got involved with machine learning, they were just classification methods. So the first one, I'm gonna very briefly describe them. This is a collaboration with Susanna Burivalova in wildlife ecology. We have sounds of rainforest, recordings of sounds. And from the recordings, we want to estimate biodiversity. And in between that, we can estimate how many species live there and in which quantities. Because a forest that is very healthy, you would expect to have not just a lot of different species, but also insufficient frequencies. You don't want one species to dominate, you know, the whole forest. You want them to have like even frequencies. So from the recordings that we actually consider them as spectrograms, images, we were able to train a neural network to classify or predict the number of species and the quantities. From that, this paper has already been published. And the ultimate goal for this project is if there are inspectors on the rainforest that they could on the fly, there are the recordings in the rainforest, they could download the recording and then run this automatic pipeline to get an just in time prediction of biodiversity. So that is where this is going. This is just the first step. There are many challenges. One is you need experts to label the recordings. So Tatiana, she is the expert in sounds. So she had to listen to the sounds and tell us the number of species and quantities for us to train the machine. The machine cannot train something that the human has not already labeled before. So that is the bottleneck of this project to start. There are some what's called self-supervised techniques that we need to start to explore, but this first step was fully supervised, so we needed label data. That is, we needed a human to give us the recordings and the quantities. The second project was a different type of data. The data were genomes, and we were trying to predict whether different bacterial strains were resistant to different types of antibiotic based on the genetic data. And then the third project was another type of data from soil, whether we could predict the potato yield or the disease susceptibility of potato from soil characteristics like microbial communities and chemical conditions of the soil. All of these three are just neural networks that are meant to classify something or predict something. And again, the bottleneck of all of them is I think is the same as you will find in biological applications. That is that you need someone to label the training data. And this is not the case. So machine learning originates in language, right? In human language, either by images or text. There's a lot of that in the internet, and you do not need experts to identify dogs from cats, right? That can be something that is done as a crowdsourcing endeavor. But you do need experts if you want to classify how many species of insects are in a recording. So I find that to be one of the biggest bottlenecks when using machine learning in biological applications. Anyway, these are all classification, but my main interest is phylogenetics. So one of the things that I wanted to explore was whether we could use machine learning techniques to estimate trees from genetic sequences. And I think maybe some of you, if you were here, so Shudong, a student that was working with me, presented in Evolution last year. He talked a little bit more about this, but I will do a summary. We saw in 2019 this paper that, to my knowledge, was the first one to try to use neural networks to estimate trees. And it could only estimate quartets, so only trees of 4 taxa. So it was like very not exciting. But they showed that it would outperform some of the traditional phylogenetic techniques like neighbor joining, parsimony, and even maximum likelihood for the case of long branch attraction. The original paper had one problem, and that is it was not accounting for tree symmetries. So if we look at the quartet here, if I swap the 1 and 2, I get the same tree, right? Or if I swap like 1, 2 to one side and the other is the same quartet. So there are tree symmetries that we need the model to account for. And the original model was not accounting for them. The only way they would deal with that was by creating all possible permutations of data and use those as training samples. So if my data, these were my 4 sequences, they will get all 24 permutations of them. So they will change the order of the rows in these 4. There are 4 factorial, there are 24. And those would be my training samples. So they will expand the training data to include all permutations. And then I talked to Leo. Leo was an assistant professor in math. He now got poached by Google, by the way. And I never heard from him again. So the last thing he told me was, I thought I was busy as assistant professor. I didn't know what busy was once he started working for Google. But anyway, he's making good money, I guess. So when he was still at UW-Madison, he's the expert in deep learning, not me. He and I, we came up with this model that was intrinsically accounting for the symmetries. So you didn't need to expand your training data set with all permutation. You just include single set of sequences, and then the model internally was invariant to the permutations. But this was still only estimating trees of 4 taxa. So again, not super exciting. Shudong worked on an extension to 5 taxa, but the bad news is that it never outperforms neighbor joining. It never outperforms neighbor joining or maximum likelihood. So you train your neural network for like 6 hours, and then you do worse than neighbor joining that takes less than a second. So it was not exciting. I gotta say, it got published because the model is actually a very good model mathematically, but it's not practical at all. Like no one is gonna use this model to estimate trees of 4 or 5 taxa. So this is one of the slides from last year when Shudong presented here in Evolution, because the date is here, September 2023. Here, what he's saying is that the rate of increase of tree space is very fast, and the number of labels equals the tree size. So for the previous case, when we have only 4 taxa, like I told you, there are only 3 possible trees. So there are 3 labels. We're classifying among 3 labels. So we're saying this sequence comes from tree 1 or tree 2 or tree 3, 3. Or if there are 5, for the case of 5 taxa, there are 15 labels. So you want to classify your sequences, which one of the 15 labels it corresponds to. But if you have 10 taxa now in your data, you have over 2 million trees. So you're trying to train a machine to distinguish between among 2 million labels. There's no way to train a neural network with that big number of labels. You would need a massive amount of training data set and a lot of time to be able to distinguish. So classification, we were actually very strict, saying that this is probably a dead end in phylogenetics. You know, this sounds very defeatist, but I really don't think that classification models are the future for phylogenetics because of how large the sample of the number of labels, how big it grows as you increase more taxa in your data set. And no one wants to infer trees that have just like 5 taxa. You know, we can do that very easily with maximum likelihood. So we think that classification models are not the way to do it. But Shudong really wanted deep learning to work in phylogenetics, so he's really investigated other type of models. He spent a year. He was a master's student, by the way. He started his PhD in Northwestern. So he spent like a year exploring different models, like the generative models. I don't know if you have heard of this website called Connected Papers. That is a website for citations. So each node in the network corresponds to different papers. I should actually include a slide here. It's actually a very good website if you have not checked it, if you want to find citations from different papers. Well, Connected Paper uses generative models for building the networks. And it's actually very common in other different graphical domains, molecules, chemical networks, social networks. They all use generative models to build networks. So it actually has a lot of success in other areas. Spoiler, it doesn't work in phylogenetics, but you don't really wanted it to work. And the way it works is that you have a sample of trees with associated sequences, and then you want to learn a distribution of topologies so that for the next sequences, you generate a tree from your distribution. That's why the name is generative. You're generating it. You're building the network rather than classifying between different options. You are building the network as you go. And you don't try 2 different ways. So one is it's called a one-shot model that is motivated by graph attention networks that were very successful. I think these were using social networks. I'm going to spare you the details, but one of the key things why we think this doesn't work in phylogenetics, and this is true for many other tools that work well in other graphs, but they just don't work in phylogenetics, is that if you have, for example, a social network, each node is a person, you have data for every single node, right? You have data, every node that is in your network, you have data for them. In phylogenetics, that is not true. We only have data at the leaves, right? We only have data at the leaves. All of the internal nodes, they don't have any data. So there is a very clear missingness pattern that is not the same for any of the other graphical domains. So we can impute the data. So if we have the sequences of the nodes, we can try to predict what was the sequence for the ancestor. But then by us doing this imputation process, we're really not learning anything new from the neighbor relationship of parent-descendant because we are imputing that sequence ourselves. Whereas in the other, each node has its own data. So from that data, we can learn neighbor relationships. So we think this is the reason why some of these generative models that Shudong tried, they all require data in every single node. We had to impute sequences in the internal nodes, but we cannot be like, information doesn't appear out of nowhere. We are using the exact same information to impute the internal nodes and then to learn the network relationship. So didn't work. I'm not gonna continue exploring that phylogenetics unless there are students that are really motivated, so you don't graduate and left. So I honestly don't know necessarily if I want to build a neural network that can build trees or networks. I don't know if that is where my passion is, but there are 2 areas where I think there is a place for machine learning in phylogenetics and in my heart. So I will tell you about that, and that has to do with embedding. And I told you that as a statistician, I really like how do we represent things as numbers in a smart way. And this paper came out in 2019 where they have a variational autoencoder. So that's a very big word. So here we have protein sequences that are aligned, and they are compressed into a vector of just two numbers, Z1, Z2. Each, it doesn't matter how long it is. So this is L. The protein sequence is length L. Each sequence is compressed as a vector of just two numbers, Z1, Z2. And this model is trained so that from that number, I can recover the original sequence. So the idea is that this vector of just two numbers contains all the information from the sequence because I can recover the original sequence with very good accuracy. So that is what autoencoder is doing, compressing something into a vector of smaller dimension. And with that vector, I can still recover the original object with high accuracy. So they're doing this for protein sequences of, this is simulated data. So we have sequences here at the tips in yellow. And then we also have sequences, because this is simulated data, we have the sequences at every internal node. So they use different colors for the sequences at every node. And here we are plotting the two vectors, the Z1 and Z2. So this is a two-dimensional plane where each of these points corresponds to a sequence. What is interesting, I think, is that there is some geometric shape in the space. So this is called a latent space, the space of this vector of just two entries. If you look at this, the yellows are at the tips. And then as you go towards the center of this cross, you are moving towards the ancestor. You see, so there is some structure in latent space that is kind of preserving the phylogenetic signal of the sequences. Like there's no reason why they should, because we are not including any phylogenetic information in the encoding. But by the way that these vectors are being created, they preserve phylogenetic signals. So this space is still reflecting phylogenetic signal from the sequences. This is with simulated data, but it's also true with real data. So this is another paper where they try different type of autoencoders. I'm just gonna focus on the variational autoencoder. I think I have a bigger image here, yes. This is again latent space. All of these papers are compressing into a vector of just two entries. I feel that it feels like black magic. Like how can you compress a sequence of 1,000 sites in just a vector that has two numbers and then be able to recover back the sequence, just two numbers? It just feels really impressive for me. But also, when you plot this space, it preserves the tree. So they are drawing the tree on top of the points, because the latent space is preserving the phylogenetic tree, the geometry of the phylogenetic tree. So what we're trying to say here is that there is a space of lower dimension that is preserving phylogenetic signal. So we don't need to work on this massive space of thousands of sites. We could actually be working on this smaller space that has just two dimensions, and it's still preserving phylogenetic signal that we want. So one of the things that we're doing right now in my lab is we're trying to use these variational autoencoders to reconstruct ancestral sequences, ancestral protein sequences. So the pipeline, it goes from, we have sequences, we train an autoencoder, then we encode all the sequences at the tips. So we have numbers. We treat those numbers as traits. We reconstruct the ancestral trait, and then we decode back to have a sequence. The main advantage of this pipeline is that we do not assume independence of sites anymore. We can do ancestral reconstruction of sequences. We have done that for many years or decades. All of the methods that we have assume that sites evolve independently, which we know that might not necessarily be true. Some might work very well in many cases, but there could be cases where that assumption doesn't work. So this new pipeline no longer assumes that the sites are independent. In fact, by the way in which we are building this vector, we are accounting for interactions between sites. The main challenges that we are dealing with is that we're using this pipeline in collaboration with two labs, Paul Alquist and Orlira Contontrafara, and we want to actually take the ancestral protein and test it in the lab. I don't do anything of that. My collaborators are going to be doing that. But it sounds exciting. You know, as a statistician, you rarely validate what you're estimating, right? You say like this is the estimate, that's it. But for this particular project, like we have an estimate for the ancestral protein sequence, and we can test in the lab if it's doing what it's supposed to do. So there is a very high bar for validation because it will either work or not work. We're working with some very simple proteins for like fluorescence that people have reconstructed them with other means. So we're first using them as a sanity proof for this pipeline. So we're very excited. And then the next thing that we want to push forward is, again, just reiterating that what I like a lot is how do we represent things that are not numbers as numbers? That is like kind of like the theme of what I do. We're trying to do that with sequences, but let's do that with networks as well. So if we are able to do the same pipeline, we could reconstruct ancestral gene interaction networks, for example. So we can encode gene interaction networks into this latent space, treat them as numerical traits, and then reconstruct what would be the ancestral one. So this is some work that is a pending grant, but we are trying to recruit students to work on this type of tools. So this is kind of like, despite me not liking machine learning, I ended up doing a lot of machine learning. So I don't think necessarily, unless it's through collaborations, that I will pursue necessarily classification tasks or inference of phylogenetic trees or networks. I mean, I'm still open to collaborating in those areas. But for me, the most exciting things is how do we embed? Embed is this process of going from something that is not a number into numbers. How do we embed things, biological objects, in a mathematical space that is easier to work with and that preserves the signal that we need for whatever inference we try to make? Okay, like I promised, this is not half microbiome. It was just going to be a quick one slide, and then I will pause to have some questions. Okay, so actually that is just the summary. So I'm trying to find connections among microbes, environments, and phenotypes. And some of the work that we have been doing has been either finding connections between microbes and the environment. And I think the next slide is probably better. Yes. So we are building what's called chain graphs. So these are networks that have two types of nodes: predictors from the environment, temperature, humidity, whatever, pesticides, and then microbe responses. And we build a network that looks like this, where we have environmental nodes as triangles and circles as the bacterial microbes. This is an R package developed by a student called Yunji Chen. Right now, we have this work for a snapshot of your data, but what we are doing as a future, we're extending as having time series of chain graphs. So if you think of this as soil microbiome, so you collect data from your soil, you build your microbial community and how it's affected by pesticides that are maybe like put into the field at time zero. And then you also want to see how the microbial community is affected at different time points. So we have two types of projects, that one is in soil with Paul Koch's lab, and one is Lake Mendota with Kartik's lab. Okay, I want to leave time for questions. So thank you. This is an acknowledgement from everybody that has worked with me all of these years. Thank you.